welcome to the International Alliance for Phytobiomes Research webinar series. We're very happy that you have joined us today. We have a very unique webinar, a little bit of a difference from our other scientific webinars, but this is going to be very exciting and very challenging for us today. So I'm really excited about our topic and our speakers today. My name is Dusty Gallagher, and I'm the Executive Director of the Phytobiomes Alliance and Project Manager of the U.S. Culture Collection Network, and this is a joint webinar series today. So just a little bit about the Phytobiomes Alliance before we bring our speakers to the screen. We are a non-profit consortium of industry, academia, and governmental scientists, and we're here to coordinate research on behalf of the full plant-based system. So our vision is that by 2050, all farmers will have the ability to have a handheld tool in the field to be able to make the best combination of decisions, what to plant, the management practices to use, and inputs for a specific field in a given year. And this information would be backed by predictive and prescriptive analytics based on geophysical and biological conditions. So we really want to provide the best opportunity for farmers to produce the most sustainable and reliable food, feed, fiber, and fuel. So we wouldn't be here today without the generosity of our sponsors. So you hear on the screen a number of entities and organizations who have come together to support the Alliance. And so we really appreciate their contributions, both in financial and leadership that they bring to our consortium. So as I mentioned in our vision, we are focused on the phytobiomes. That's much broader than any microbiome. And so we define the phytobiome as a complex system of plant-based agriculture. So phyto meaning plant, biome meaning a specific site. In this particular infographic, we show soil, but it could be hydroponics, it could be pasture, it could be controlled environment agriculture, it can be many different forms. But we are looking at all of the interactions that occur in and among all of the environment that plants are grown. So we've got plants growing in soil, we've got the microbiomes, microorganisms, macroorganisms, we have other animals and plants that influence the system, and then we also have climate, weather, and water. And all of this is impacted by and influenced by the management practices that are chosen by the agricultural producer. So this entire system is what we're trying to understand and unlock all of the interesting and complex interactions that occur. And one of our projects through the Alliance is the U.S. Culture Collection Network. And this is funded by the National Science Foundation through their Research Coordination Network program. And so through this program, we're trying to bring together scientists that work with living microbial collections, because this is one of the key pieces in that plant-based system of how those microbes interact with and among each other and what impacts that has on the plant growing. So this is why the Phytobiomes Alliance is partnering with the USCCN and NSF for this virtual coordination network. So one of the major focuses of USCCN is to build and create a public registry and database. So we have an online tool that shows a census of microbes, where those collections are, what types of collections, whether they're research collections, federal collections, what is available and where. Do they have genome sequences associated with those microbes? And it also, probably most importantly, tells you who to contact. So with this tool, we're trying to build those collaborations between research scientists and where these very important living microbes are, mainly in the US, but we also have some international participants in this registry. And we are culminating our work through USCCN by holding a conference, and it's actually coming up very quickly next week in Providence, Rhode Island. We will be holding a one-day conference to really talk about what the USCCN has accomplished over the last few years, and most importantly, where USCCN needs to head over the next few years in terms of supporting those microbial collections. So as most of you know, we have a full Alliance webinar series, and these are just a couple of the upcoming ones this fall. So in September, we will have a focus on soil health and carbon cycling. And then in October, we'll focus on our animal health and animal microbiome linkages with looking at animal and grassland microbiomes and the impact that manure has on those systems. So the links are available. The registration is open, so make sure that you plan to attend those upcoming webinars. And finally, I might just say later in 2026, in November, we will have the culminating International Phytobiomes Conference. And so we will be at Niagara-on-the-Lake in Ontario, Canada. And this is a larger conference. We have over 3 days. We bring together the latest and greatest in phytobiomes research and talk about where we're headed with that phytobiomes community in the research in the coming years. And so registration is open, and there are a few upcoming deadlines. If you're interested in submitting an abstract, that occurs later in August 28th. And then the early bird registration is shortly after that, ending in September 4th. So we would love for you to join us in Canada for this conference. It's a great way to network and to understand what's happening in the phytobiomes community and with phytobiomes research. So here are just a few announcements and logistics for today's webinar. The webinar is being recorded and will be posted on the Phytobiomes Alliance YouTube channel for you to watch, and that should be available later today or tomorrow. And so if you want to go back and catch a certain section of this webinar, then you're able to go and view that. And then we have a little bit of a different panel that you can participate in. Following our three presenters, we will have the opportunity for an open Q&A. And so there is a very specific Q&A panel on your Zoom webinar application. And so we ask that you submit all of your questions in that panel. The chat that most of us are familiar with has been disabled for all of the participants, but it's a way for the organizers to communicate with you. So we will be posting just information about today's webinar, links that you will see in certain presentations, any PDFs that are relevant to the presentations will all be posted in chat or handouts. So again, if you would like to ask a question to our speakers, the Q&A panel is where you should go to do that. And without further ado, I'd like to introduce today's webinar. We're going to be talking about navigating the regulatory complexity of microbial interventions for climate change mitigation. And so I'm very happy to introduce our speakers for today, Matthew Ryan, who is from CABI in the UK, Keith Matthews, who is a principal at Matthews Law here in the US, and we have Belén Guijarro, who is from Spain, and she will be discussing the EU. So again, we're very excited and we're so happy that these speakers are able to join us. And I would like to invite Matt Ryan to the screen, and he will begin our presentations.
Thank you, Dusty. And before we hear from our experts, today. I'd just like to thank the Phytobiomes Alliance for hosting and to Keith and Belén for their time today. So, what is the Microclimate Project? Next slide, please, Dusty. The main goal is to understand how microorganisms, plants, and soils interact in the context of climate change, with the eventual impact of strengthening sustainable agriculture and forestry and support biodiversity conservation under climate change conditions. What's unique about this is the M4C approach, and that's to provide access to advanced research infrastructures and to provide expert support through training opportunities and other services for our users. Next slide, please. To give the project some context, it is a rather large EU project worth nearly 15 million euros. It is coordinated by Anna Portugal, who's based in the University of Minho in Braga, in Portugal. And it's an InfraServe project. Next slide, please. It has 31 partners. And importantly, it involves 4 EU research infrastructures. They are MIRI, who are the coordinator, Emphasis, LifeWatch Eric, and Anna Eric. In addition, there are 2 international partners, and they are my organisation, CABI, a UN treaty level organisation focused on international development, and the International Alliance for Phytobiomes Research, who are hosting us today. Next slide, please. The main project goals are centred around 5 key themes, which effectively involve access to services, integration, training, and engagement. And together this works for the benefit of the project and the wider European infrastructures. Next slide, please. To better describe this, we need to look at how the project operates. And you'll see in the middle there's a transnational access programme. And that is a facility to allow users to access the resources and infrastructure of the member participants of Microbes for Climate. But in the context of today, it relates to work package 6, which is the legal, regulatory and intellectual property aspects of how we take microbial solutions and apply them to mitigate against climate change, whether they be single xenic organisms or more complicated microbial communities. That work package has 3 tasks: bioethics, led by the Austrian Institute of Technology, intellectual property, which includes patent law, led by the University of Minho. And what's interesting with patents at the moment is you can only really deposit single strains rather than complex defined microbial communities. And then task 3, which relates to this seminar today, which also includes the Nagoya Protocol and ABS legislation. And one of the outputs of the project is a paper, Policy in Practice, How to Do the Nagoya Protocol, which was produced jointly with the Micro Project. So do have an opportunity to look at that, but that's not the focus of today's talk. Next slide, please. One of the early things to come out of discussions that we had was the unfortunate terminology syncoms. There's nothing synthetic about a microbial community. So a group of international researchers has proposed a realigned term, defined microbial communities, to facilitate clearer terminology, which may be helpful for regulatory purposes. Next slide, please, Dusty. And this is the problem. There is so much confusing terminology, and this does lead to some regulatory ambiguity. One person's bioinoculant might be another person's biostimulant. And an interesting example, just last week, I was at a conference in London where a researcher had used a biopesticide called metarhizium and was investigating its plant protection properties. That example alone will then lead you to ask, well, what would that be registered under current legislation? Would it be a biopesticide or perhaps a biostimulant or even a biofertilizer? Ultimately, clarity is absolutely crucial, and we need to work towards that. as scientists. Next slide, please. And that brings us to what today's webinar is about and what potential questions we may ask. We need to begin to untangle the complexity of environmental regulations for end use. For example, why using a defined microbial community might be a lot more complex than using a single exenic organism. We may wish to provide an oversight of different approaches to environmental regulation in different jurisdictions. And with Keith and Belén today, we'll get a take on differences, for example, between the US regulatory environment and the EU regulatory environment. But a key question is, what should a researcher do when developing a potential microbial solution? There are so many research groups at the moment developing defined microbial communities, but they rarely think of how they might eventually get this approved for eventual usage. And there's so many things that might help that easier, including the terminology. And the other key thing to consider is how the regulatory landscape will not only change, but what it might look like in the future. So without further ado, I will hand over to Keith, who will take us through things from a US perspective. Keith.
Thank you, Matthew. And good morning, everyone. Good morning to everyone who's in the United States. Good afternoon, good evening to those around the world. I'd like to echo Matthew in expressing my sincere thanks and appreciation to the Phytobiomes Alliance for allowing me to participate in this webinar this morning in the US to speak about the regulatory system in the US for the type of microbial organisms that Matthew just spoke of. And I will say that his last words were an excellent segue into my presentation. So let me start out by saying that we've agreed to keep our presentations to about 15 minutes or so. And so that's an extraordinarily short period of time to speak about the United States regulatory system, which is extraordinarily complex. I urge everyone to pay particular attention not only to my presentation, but also to Belén, who will follow me, because I would posit that the regulatory approach in the EU is a bit more straightforward than in the United States. The regulatory system in the United States is exceedingly complex, and it's grown up over a number of years. And it's kind of like the animal brain, the evolution of the animal brain. Just keep getting pieces put on top of existing pieces, and so eventually you just have a very complex system. So what I'm going to focus on this morning are the regulatory approaches at the United States Environmental Protection Agency and the USDA. There's also another agency that may, in fact, have some involvement with the type of microbial organisms that we're talking about. That's the the Food and Drug Administration, FDA. But I'm not going to focus on FDA right now, just again, because of time limitations. So we can go to the next slide, please. So I particularly liked the graphic that Matthew put up regarding the different terms. And I will say at the outset, I think both Matthew and Belén have a beautiful set of slides. They have a lot of graphics, really visually, arresting and pleasant. My slides are just words. For me, if I were an artist, my medium would be words. Words matter, particularly in the U.S. regulatory context. Words matter. And if you think about, if you look at what I have on the screen here, this is a description of the webinar that was on the Phytobiomes Alliance emails that they've been sending out. And it really does kind of dovetail very well with the point that Matthew was making about the different terms. Biopesticide, bioadjuvant, biofertilizer, biostimulant. Are they different? How are they different? In the U.S. regulatory system, how are they different types of these different types of products that may be defined or spoken of using these different terms? Are they regulated differently? A biostimulant can be both a biopesticide or a non-biopesticide. That will have implications for how it's regulated. A biofertilizer or a bioadjuvant, they could be the same thing. A biofertilizer could, in fact, be a biostimulant, and a non-biopesticide biostimulant. As I said, fairly complicated, but let's jump into this and start with the next slide. So in the US, biopesticides are a category of pesticides. Pesticides are defined as any substance or mixture of substances that's intended for preventing, destroying, repelling, or mitigating any pest. The key point here is that it's not the substance itself. A substance is not a pesticide merely in and of itself. It is a pesticide because of the intended use of that substance. Why is it being sold and distributed? If it's being sold and distributed with the intent to prevent, destroy, repel, or mitigate a pest, then it's a pesticide. Also, so the definition of pesticide in the United States has two prongs. Second prong is it intended for use as a plant regulator, defoliant, or desiccant? We're going to focus on plant regulator here. Next slide. So what is a plant regulator? It's any substance or mixture of substances that are intended, there's that word again, intended through physiological action to accelerate or retard the rate of growth or maturation of a plant, or for otherwise altering the behavior of plants or the produce thereof. There's no other regulatory system in the world that has this sort of a definition for pesticide that regulates these sorts of chemicals as pesticides. But if you have a substance and the intent for the use of that substance is to alter the physiological behavior of a plant or accelerate or retard the rate of growth or rate of maturation, that's a pesticide. I can go into why that came into US law, but we don't have time to discuss that. But this just suffice it to say that that's within the definition currently. Now, that definition excludes certain substances. Of interest to us are plant inoculants. Next slide. What's a plant inoculant? EPA defines a plant inoculant as a product that consists of microorganisms that are intended to be applied to the soil or to the plant for the purpose of enhancing the availability or uptake of plant nutrients through the root system. So if that in fact is the purpose of your microorganism, then your microorganism will in fact not be considered a plant regulator and will not be regulated as a pesticide in the US. So next slide. So we're not done with EPA yet. So that regulatory program was under the Federal Insecticide, Fungicide, and Rodenticide Act, FIFRA. EPA also regulates microorganisms under the Toxic Substances Control Act, TSCA. And how do those regulations differ? Well, first of all, if your intent is for a microorganism to be used for a pesticidal use, either to prevent, destroy, repel, or mitigate a pest, or as a plant regulator, you're not subject to TSCA. Other uses, however, such as industrial uses, you may in fact be subject to TSCA because TSCA regulates chemical substances. Definition of chemical substances, any combination of substances occurring in whole or in part as a result of a chemical reaction or occurring in nature. Pretty broad definition. Living microorganisms fall within that definition. So let's go to the next slide. So very briefly, EPA imposes regulatory requirements under TSCA on a specific category of microorganisms. Those microorganisms that are genetically engineered and that are transgenic. I'm sorry, not transgenic, that are intergeneric. So if you have a microorganism that is intergeneric and has been genetically engineered, it is subject to regulation by EPA under TSCA. Microorganisms that are not intergeneric or that have not been genetically engineered are not regulated under TSCA. So next slide. So there we're done with EPA. But we still have to consider whether or not a microorganism might be regulated by the USDA under the Plant Protection Act. And I should note that I intended to note this at the very beginning. These regulatory systems, EPA, USDA, FDA are not exclusive. So you can have a microorganism that is subject to regulation by any of the three, all of the three, or some combination of the three. So under USDA regulates under the Plant Protection Act, which authorizes USDA to control or prevent the movement or environmental release of plant pests or biological control organisms. Definition of a plant pest is any organism that can directly or indirectly injure or cause disease or damage to any plant or the parts thereof. So USDA's focus is very narrow as opposed to EPA's focus. Next slide. The way that USDA regulates, however, is through under PPA. They implement PPA through the Animal Plant Health and Inspection Service, APHIS, which has two sub-organizations, Plant Protection and Quarantine Biotechnology Regulatory Service, PPQ and BRS. Plant Protection and Quarantine regulates conventional wild-type microorganisms. BRS will regulate genetically engineered organisms, including genetically engineered microorganisms. I will talk, let's go back. I will talk a bit about some of the changes that seem to be in the offing for USDA regulation of microorganisms by PPQ and BRS, one of which is to try to conform the regulatory approach of the two sub-agencies. It's kind of interesting that over the years, they're separate. And so I guess as being separate, they have developed regulatory programs that are somewhat distinct. And so you have a particular regulatory approach if you are producing and subject to regulation for a conventional microorganism, and a somewhat different approach if you are dealing with a genetically engineered microorganism. The two sub-agencies, PPQ and BRS, are actually working now to try to bring some conformity to their regulatory programs. And as I will discuss very shortly, that may be a bit more overarching and comprehensive change to other than the two agencies seeking to coordinate their processes. So next slide. And that results from the APHIS Secure Rule. The Secure Rule is basically amendments to 7 CFR Part 340, the BRS regulatory program that was promulgated in 2020 and implemented a fairly comprehensive update of the BRS regulation. It can be said that the primary purpose of the Secure Rule was to revise the APHIS regulations applicable to GE organisms, genetically engineered organisms, to make those regulations more fit for purpose, given the technological advances that had occurred. It had a very long history. USDA first proposed update to Part 340, I believe, in 2007. Went through a number of different iterations, a couple of additional proposed rules. Finally ended up promulgating their final rule in 2020, and it was then vacated by court order in 2024. Very typical of the US regulatory system. So what in effect happened was APHIS updated their regulations in 2020 to make them more fit for purpose. That regulation was vacated in 2024. Now APHIS is effectively implementing a regulation that is circa 1987. Next slide. However, to address this, and I'm coming to the end of my, my talk here, but to address this, APHIS is moving forward. USDA actually is moving forward. And in May of this year, USDA published a request for information on its regulatory program, how to move forward with a new regulatory scheme given where we stand now. And so I urge you to this is the quote there. This is a quote from the APHIS request for information in the Federal Register. And this basically stated what they, in fact, are trying to do. They're trying to consider alternative frameworks for the regulation of genetically engineered organisms under the PPA to ensure that regulation and oversight are proportionate to risk. Biotechnology developers in the United States have a predictable regulatory pathway to commercialization under the PPA, I might add. That would be solely under the PPA. And the next one, I don't know. I guess they felt like they had to put this in here to ensure that APHIS operates within its statutory authority. APHIS has broad statutory authority. I don't think, at the end of the day, I think it's hard to say, to conclude that they would be operating, outside of their statutory authority. In fact, the secure rule, as I said, was vacated by a court on the basis that the court felt that APHIS had not fully implemented its statutory authority. The court didn't say, well, we think you did stuff, we think you did something you weren't authorized to do. The statute doesn't allow you to do that. What the court, in fact, said was statute requires you to do more than you did. And therefore, I'm going to, as a judge, as a court, I'm going to vacate this rule. So next slide. And these are specific questions that that APHIS USDA asked in their request for information. There's 8 of them. I'm not going to go through all 8, but I will say that there are some that are very key. And the first one, that begins with the first one. Are there material differences in plant pest risk between conventional genetically engineered organisms? Quite frankly, it has been the policy of the United States government since 1986. It's been the stated policy of the United States government since 1986 with the issuance of the Coordinated Framework for the Regulation of Biotechnology that no, there aren't material differences in actual risk between conventional and genetically engineered organisms. Notwithstanding that, the regulatory program in the United States over those 40 years has been to regulate as if there were. The Coordinated Framework says the policy of the United States government is to regulate on the basis of product, not process. For 40 years, the United States regulatory agencies have regulated genetically engineered organisms on the basis of process. It would be nice if we could actually bring to fruition what the government said they would actually do 40 years ago. And I will close out here, but I will just point out that, so there's some other questions here that were very descriptive, and I think were intended to be, for those commenters that provided useful, substantive responses to these questions. These were intended to form a basis, to provide USDA with a basis for going forward with a potential update to their regulatory program that, in fact, would have a rational basis. So, Dusty, if we could just look at the next slide very quickly. So this is the questions 5 through 8. And so, these are the questions that USDA really asks for substantive input on to help them formulate, develop, and then potentially publish an alternative regulatory system for genetically engineered organisms under the Plant Protection Act. And I will stop here, but I will say that the comment period ended. I've looked at a number of comments that were submitted, really have a lot of admiration, and want to say that there were a number of commenters that provided really outstanding substantive input to these questions. And I do think that the USDA process has been substantially benefited by a number of comments that were submitted in response to this request for information. And so with that, I'm going to close out and turn it over to Belén.
Thank you. Thank you, Keith. Well, good morning or good afternoon, depending on the time slot you are in. And thank you for the opportunity to share with you or work on the risk assessment for microbial active substances and novel microbiome solutions under EU legislation. Next slide, please. After Kate and Matthew talks, it's clear that microorganisms offer an enormous opportunity for sustainable agriculture. However, unlike synthetic chemicals, microorganisms are living organisms, meaning they reproduce, evolve, interact with plants, with other organisms, with other microorganisms. So the thing is that for many years, Europe assessment microbial products by using the same framework originally designed for chemicals. And now I think we are trying to change this situation. Today we are moving away from the simple adapting chemical approaches, and now we are trying to implement our risk assessment schemes specifically designed for microorganisms, where biological understanding becomes the starting point of regulatory decision making. Next slide, please. So European legislation, as we all know, is solid. We have Regulation 1107/2009 and the related data requirements and uniform principles, Part A for chemicals and Part B for microbials. And all active substances have to meet the same fundamental requirements to be authorized. They must not cause harmful effect on human health, animal health, or the environment. And these principles applies equally to chemicals and microbials. However, microorganisms raise different scientific questions requiring different type of evidence. And these recognitions led to an important regulatory update. Next slide, please. In 2018, the European Commission launched an expert working group involving experts from all member states. I participate in this process to revise the data requirements and the uniform principles for microbial active substance and plant protection products. The idea was not to create a separate pesticide legislation for microbials, meaning that Regulation 1107 remains the central legal framework. However, in 2022, the Commission amended the approval criteria, the data requirements, and the uniform principles to make them more specific for microbial active substance and their products. Next slide, please. The updated data requirements are structured around the biology of the microorganisms. This is a key new element. So the new frameworks require us to characterize microorganisms in much greater detail, taking in consideration deeply the identity, ecology, the mode of action, pathogenicity, infectivity, antimicrobial resistance, secondary metabolite of potential concern, all become central components of the assessment. And these are no longer isolated requirements one by one. Together, they build the scientific evidence needed to determine whether a microorganism might realistically pose a risk, and this is very relevant, under the proposed conditions of use. Next slide, please. So one of the most important messages I would like to emphasize here is that registration starts with microbial identity. So identity is much more than assigning a species name. It means demonstrating undeniable which strain is being assessed, ensuring traceability through the culture collection deposition, confirming identity through the whole genome sequencing, understanding its origin, and guaranteeing consistency during the manufacturing process. So without a robust identity, the entire risk assessment becomes uncertainty. It's the core first step of the analysis. Next slide, please. One of the most relevant, interesting, and challenging modification under my point of view was that for the first time, the new data requirement explicitly allow the approval of an active substance consisting of a qualitative defined combination of strains. In other words, a microbial consortium. And this clearly reflects the fact that innovation is moving beyond single strain products towards microbial consortia that have complementary functions and might improve efficacy under variable environmental conditions and climate change. However, regulatory complexity also increases in these consortia and active substances, and the assessment must address strain interactions, manufacturing consistency, stability over the time, and whether the evidence should be generated from each strain or for the consortium as a manufacturer or both. So under the 2022 legislation, when the active substance is a consortium, the definition of microorganisms apply to the consortium as a whole. And here we will deal with different types of consortia. We have defined synthetic community, or as we call SYNCON, may be easier to assess because their members can be identified, can be characterized and deposited individually. We also have natural communities and enrichment cultures that are more challenges in terms of legislation and risk assessment because some members might be difficult to isolate or maintain separately and many other things that come up. So the central regulatory question here is whether the consortium can be assessed as a functional biological unit or whether components level assessment is also required. Next slide, please. Several European projects are currently helping address these questions. One example is the Ration project funded by Europe Horizontal project, where we are developing a new risk assessment approaches for microbial pesticides, microbial consortia, and other biological low-risk products. The objective is not simple to generate more data, but to develop a practical regulatory tools and guidance that can support future authorizations. So Ration project, risk assessment innovation, we are 29 partners from across Europe, industry, academia, and regulatory bodies representing 13 countries. This mix of expertise allows us to combine regulatory insights, scientific innovation, and practical industry experience. And it focuses on three main products, microbials, plant extracts, semiochemicals, and RNA-based products. I will focus here in the microbials. Next slide, please. Within Ration, in Work Packet 2, we have focused on 4 complementary areas for the risk assessment of microbial active substance. First, we are developing a whole genome sequences pipeline to support regulatory decision making. Second, we are evaluating whether current ecotoxicological methods are appropriate for microorganisms. Third, we are validating the approach using 3 representative microbial case studies, a bacterium, Bacillus belenenses, a fungus, Purpuricilla liliaceum, and a baculovirus. And finally, we are extending this concept to emerging technologies such as microbial consortia and protists. Next slide, please. The whole genome sequence is a key element for the evaluation of microbial active substances, and it's becoming one of the most powerful regulatory tools. It supports strain identity, antimicrobial resistant assessment, detection of virulence-associated genes, prediction of secondary metabolite biosynthetic clusters, and traceability. Importantly, the whole genome sequence is not intended to replace a traditional study. We already need phenotypic approaches. Instead, it provides an early tier decision support tool that help focus subsequent investigations where they are really needed. In the framework of the project, we have developed a new open access pipeline which integrates a series of up-to-date software offering first a high accuracy in strain identification, detection of antibiotic resistance genes, the location in the genome and the potency of transferability, detection of pathogenicity potency, human pathogenic through the identification of virulence factors, detection and description of gene clusters associated with biosynthesis of secondary metabolites of potential concern. Next slide, please. So beyond single microorganisms, we are also studied different microbial consortia in order to develop a new risk assessment approach specific for this type of new tools. And this include combination of fungi. We have a consortium of Trichoderma and Fusarium with different mode of action against Verticillium. We also have a combination of 3 Penicillium rubens strains that acts against soil-borne pathogen, specifically for Fusarium. It's also an intra-kingdom syncom. The third consortium is a combination of 3 Bacillus species against Fusarium and Rhizoctonia, and the fourth one is a trans-kingdom syncon, and the novelty is that combine a Buschura mycorrhizal fungus, Rhizophagus irregularis, with a Bacillus belenensis. Next slide, please. The second project we are involved in is an EFSA-funded initiative called Fit for Purpose Risk Assessment for Low Concern Active Substances and Uses, and is led by Aristotle University of Thessaloniki together with Wageningen Environmental Research, the CTGB in Holland, and INIA-CSIC in Spain, and several other partners. The objective is similar: to make a risk assessment more scientifically oriented, more efficient, and more proportionate to the actual risks. And this work complements RATION and contributes also to the future European guidance for this type of low-risk active substances. Next slide, please. So this slide summarizes the direction in which microbial risk assessment is evolving. It's a combination of the results of both projects. So instead of testing everything, we begin with the concept of problem formulation, and we ask, could these microorganisms realistically cause harm under the proposed conditions of use? Then we identify biologically plausible pathways to harm. Only if these pathways remain plausible do we request additional experimental evidence, and this allows us to focus resources on studies that are truly informative while avoiding unnecessary testing. It is more scientific, I think. It is more transparent and more efficient approach to risk assessment. Next slide, please. So I would like to finish with 4 key messages. First, researchers should think about regulation from the very beginning of product development. Second, microbial identity is not simple a taxonomic exercise. It is more. It is the cornerstone of the entire regulatory assessment. Third, biology should guide the risk assessment through program formulation rather than relying on standard testing alone. And finally, European collaborative projects such as RATION, EFSA, Microbes for Climate Change are helping transform scientific innovation into a practical regulatory solution. The main message I would like to leave you is that regulation should not be considered only at the end of the product development. Successful registration begins long before the dossier is submitted. Next slide, please. So thank you very much for your attention, and I would like also to thank all people from the project and from INIA involved in the projects, David Ayuso, Maria Jose Patiño, Jose Luis Alonso, and I will be happy to answer any questions and discuss about the topic. Thank you.
Thank you all for your very engaging presentations. We're bringing everyone on screen, and we will begin the Q&A portion of our webinar. And I might just remind the participants that if you see a question that has already been posted in the Q&A panel, you have the opportunity to upvote that. So if you see something that you would like asked, please feel free to upvote that, and we will try to get to that and all of the questions that you have posted here. So let's start. Let's begin. I think this first question is for Keith. It says, "Hi Keith, thank you for your presentation. Would you be able to speak more to the regulatory registration differences of applying a microbe as a plant regulator versus a microbially derived metabolite as a product? Do purified metabolites for crop enhancement plant regulator products fall under FIFRA regulations, or does it sit in a gray area?"
No, I wouldn't say it fits in a gray area. I would absolutely say that it falls under FIFRA. It falls under BPPD, the Biopesticide and Pollution Prevention Division at EPA. And if I recall the exact wording of the question correctly, the purified extract would be considered a biochemical. So in BPPD, the regulatory scheme is divided between biochemicals, microbes, and what's known as emerging technology products. So mostly genetically engineered products. So that purified extract would be considered a biochemical plant regulator, and you would have to meet the same regulatory requirements as a microorganism or microbial plant regulator product, although the specifics of how you do that might be different.
Okay, thank you very much. Okay, Belén, in your presentation, you mentioned that active substances shall be described not only up to the species level, but to the strain level. So how do you differentiate two strains of the same species? And is there guidance about this?
For that purpose, you can use specifically the whole genome sequence analysis and compare genome sequence and identify the difference between the genomes. There are guidance documents developed by EFSA for the analysis of the whole genome sequence, on which you can identify the information necessary to analyze the whole genome sequence.
Okay, thank you. Okay, this next question I don't believe is directed toward any one panelist, but let's read it and we'll let you all respond if you would like. Typically, registration is overwhelmingly a pre-release exercise, yet a self-propagating microbial intervention disperses beyond the treated field and persists. What post-release monitoring or recall obligations actually exist in either jurisdiction, And is demonstrated reversibility ever a condition of approval, or does approval in practice assume permanence? That was a very long question. Let me know if you'd like for me to repeat it. I'm guessing either Keith or Belén, would you like to offer?
Yeah, Belén, would you like to go first?
You're muted, Belén.
Yes, please, Keith, go ahead.
Oh, okay. Well, I would say, again, the US is a very complicated regulatory system. And so that question could, in fact, be posed both to EPA and to USDA, depending upon what is the regulatory jurisdiction, how the regulatory jurisdiction for a particular microbe would actually fall. And quite frankly, the answer might be different, depending upon whether you're looking at USDA versus EPA. I will speak specifically to the post-registration obligations. Under FIFRA Section 682, if a registrant has factual information related to potential adverse effects of a registered pesticide that EPA is not aware of, they are obligated to inform the agency of that. So in that context, the way I would answer that question is, if in fact the microbe persists or has spread from the field where it was applied, does it constitute an adverse effect? Is that microbe having an adverse effect where it now is? If it is, then the registrant has an obligation to inform EPA of that. If it's not, then that obligation isn't there. And also, that may be something that was in fact addressed during the registration process. It may have been something that EPA in its risk assessment looked at. What is the likelihood that this microbe might spread beyond the application area? And is that a concern? So I'll stop there.
Okay. Belén, do you have anything to add? You would like to add?
Yes, thank you, Dusty and Keith. Yes, in Europe, it's also very complicated, the approval and the process for registration of plant protection products, starting with the authorization of the active substance. And this process is a long process on which all the member states are involved, and also EFSA is involved. The registration period is for 10 years. After that, there is a period of renovation of the active substance.
Okay, thank you. We probably just have time for one more question, but I will mention to the participants that we have an agreement from our presenters today to get the rest of the questions answered to you following the webinar. So if your question is not asked, we will make every effort to get it answered in writing. So the last question, thank you to all the speakers. There's a general question here. To any of the panelists, do you see or foresee pressure to harmonize regulations, particularly between Europe and the US, in particular as regards to GMOs? Matt, would you like to, since you have been quiet in the Q&A, would you like to offer a perspective here, or would you like to pass to either Belén or Keith?
Yeah, I would say that GMOs add another level of complexity that's not always well received by end users or those making the interventions. I suggest that Keith might be slightly better positioned to answer the question on GMOs.
Yeah, I'll just very quickly, because I know we're short on time, I'll just say that, yeah, I agree with that, Matthew. And it's unfortunate because I think that it, is a result of just a lot of ignorance and scientific illiteracy that's out there with respect to what's the actual risk of genetically engineered organisms versus organisms that are not genetically engineered or produced by non-genetically engineered processes, which is unfortunate. But you, in fact, do have a very different approach in the EU than you have in America, than the United States. I don't think that there's really going to be any sort of harmonization there, because the United States has very specific statutory and regulatory obligations, which, at this present time can't be met with a more EU type approach. And I'd be very curious to hear Belén's perspective on that as well. Either she agrees with me or disagrees with me.
Okay, Belén, you get the final word.
Yeah, I fully agree with you, Kins. Yes, the EU legislation in GMOs is very complicated, very strict, and in terms of plant protection product, it's very difficult because they have to fulfill first the legislation on GMO and then 1107/2009. So I think there is non-GMO authorized as a active substance as a plant protection product nowadays.
Okay. Well, thank you very much to all of our speakers, all of our participants. This was an excellent discussion and one that obviously needs a lot more discussion in the future. So thanks to everybody who joined us. Have a wonderful day.
Thank you very much.
Take care, everyone.
Thanks. Bye-bye.